Good morning. My name is Ashanti Anderson, and today I will be discussing with you the significance of DNA as it relates to research, medicine, and biological aspects from the graduate level. This presentation is intended for educational and teaching purposes for honors or AP high school and undergraduate science students. I am a student of Grand Canyon University. For the teaching aspect, this lesson is intended to be a precursor to an exam accompanied with a lab and provided in the lectures. The lab will consist of designing of a DNA molecule, identifying the differences between prokaryotic and eukaryotic cells, and the presenting of the information learned to the class. This will be an objective that the students will have to meet in order to fully understand and grasp the concept of the lesson. The objective of this presentation is to understand the basis of DNA, the differences in cell composition, eukaryotes versus prokaryotes, and to understand the basics of cell signaling and the biological pathways. There will be a brief overview about the biological pathways and cell signaling. It will not be an in-depth overview, though. Let's go ahead and get into this. What is DNA? DNA stands for deoxyribonucleic acid, whereas RNA is ribonucleic acid. DNA is the hereditary information of all living cells. This is the actual blueprint that creates the basis for every cell's composition versus physical and genetic wise. Without DNA, we would not be able to see the differences within each other, either molecularly or biologically, physically. DNA is composed of nucleotides, which are small units composed of a phosphate, sugar, and one of four bases. DNA is known for its double helix structure. So as I said before, DNA is the biological blueprint that provides information for all living organisms. DNA is a polymer composed of small units called nucleotides. Nucleotides consist of a phosphate, sugar, and four bases: adenine, thymine, cytosine, and guanine. These pair together, usually A with T or C with G. DNA uses these base pairs to create the structure that is their double helix structure. When these base pairs connect together, they actually create a bond that allows the two strands of DNA to interact with each other, causing a spiral corkscrew shape that you see in DNA structures. It is nucleotides that are responsible for creating the backbone that is the structure of DNA, like I've already said. Each basis component is paired to one another along the double helix. The double helix is formed by two strands that twist around each other in a corkscrew shape. A single strand of DNA is formed by chemical bonds of one phosphate of a nucleotide to the sugar of the next in a long chain of nucleotides. Basically, one component from one strand has to match up like a puzzle piece to the other strand. These all connect with each other as a puzzle piece would. So every base pair has their own unique shape, and they're fitted together perfectly so that they match up with each other and create this backbone that we see. So basically, the chemical bonds, which are hydrogen bonds between these pairs, cause a very strong attraction. The long strands of nucleotides that provide the structure are composed of chromosomes, which contain genes and many nucleotides that communicate to the cell through proteins. The spiral shape of the strands are flip-flopped, so the 3' end pairs, the 3' end right here and the 5' end right here, these numbers that you see on the ends actually tell the length of these strands and the order and position of the strands. So 3' will always match up with 5' on the top half of the strands when they meet together. On the bottom half of the strands, the 3' end will always match the 5' but in the opposite position, because when they spiral, it causes the strands to flip-flop positions. The spiral shape of the strands are flip-flopped so that the 3' end pairs with the 5' end. From a chemistry aspect, these base pairs are connected through hydrogen bonds, as I already explained before, and you can see right here the hydrogen bonds. You can see right here how the oxygen and the hydrogen, the two hydrogens are matched with the nitrogen, because the two water molecules, the two hydrogen molecules that are in water, actually come apart from the oxygen to attach to the nitrogen part, as well as the hydrogen molecule bonds with the nitrogen molecule where it needs to take place. This has to do with electronegativity. That's a chemistry aspect. We'll get into that later. But for right now, just understand that for these bonds to come together, something has to lose its place somewhere. So these hydrogens have to separate from the oxygen in order to match this bond, so that they can come together, so that these base pairs can complement one another. From a chemistry aspect, these base pairs are connected through hydrogen bonds that create a polarity of positivity that cause the linkage. As I said before, this is the attraction I was talking about, where one of these chemical compositions must lose something and be placed somewhere else in order for these components to be attracted to each other, and these base pairs to be efficient in making the DNA structure. It is through genomic sequences that we can relate biological mutations or abnormalities. Through looking at DNA, we can actually find issues that may be wrong with somebody genetically, biologically, but it is through genomic sequences, which genes are part of chromosomes, which are in DNA. These things all tie together so that we can use them to research and study things within our own human nature or animal nature as well. Let's continue. Prokaryotes versus eukaryotes. This is very important, this lesson right here, because there are different ways in which DNA functions through prokaryotic cells versus eukaryotic cells, and I figured I would touch on this very briefly. Prokaryotes experience binary fission or prokaryotic fission, which is used to describe cell division in prokaryotes and single eukaryotic cells. DNA of a prokaryotic cell is in a single linear chromosome. As you can see right here, prokaryotes are definitely shaped differently than eukaryotes, which are more circular. These prokaryotes tend to be more linear in structure, and they lack a membrane, a plasma membrane. Instead, they have a cell wall and an intermembrane, whereas eukaryotes have a membrane-enclosed nucleus, and they are known for having a true nucleus. Eukaryotes usually contain more DNA than prokaryotes, are linear instead of circular. So, because the surface area of eukaryotes is much larger than that of prokaryotes, they tend to be able to carry more material than a prokaryotic cell would. As you can recall, prokaryotes are cells that lack a nucleus, while eukaryotes are cells known for having a true nucleus and membrane-bound organelles. The prokaryotic cell cycle, while DNA is being replicated, is also responsible for prokaryotic fission. During binary fission, there are 4 steps that occur in relation to prokaryotic cells. Right here, I have provided a description of the steps and what binary fission is as it relates to prokaryotic cells. The prokaryotic cell cycle, while DNA is being replicated, is also responsible for prokaryotic fission. During binary fission, there are 4 steps that occur in relation to prokaryotic cells. In prokaryotes, DNA is surrounded by a cell membrane within a cell wall. The first step contains 1 chromosome. The DNA is then replicated and attaches to the cell membrane. Right here, DNA is replicated and attaches to the cell membrane in step 2. The new cell wall is then established, and the membrane growth between the 2 attachment points increases. This causes the cell to separate independently into two portions, as we see in the last step here, along the attachment sites. Now, the attachment sites right here, this would be, if we were looking at an animal cell, we would say a cleavage furrow, but because prokaryotes are bacterial cells, we're going to call it an attachment site. So right here along this attachment site, we see the splitting of the cells into two identical daughter cells, which are exact copies of the original cell we started off with, the parent cell, in the first step. The whole point of this is to make genetic copies and share the DNA that is within the parent cell by creating two daughter cells that could then transfer the genetic information throughout the cell, if that makes any sense. So, eukaryotic cells are the primary cells responsible for the double helix structure because they bind around histone proteins, creating the corkscrew shape. So as we see here, like I said before, eukaryotes tend to be a lot bigger surface area wise than prokaryotic cells because we have to consider the mitochondria. We have to consider placements of the nucleolus because prokaryotes lack a nucleus. Eukaryotes have a nucleus and a nucleolus, which lies inside of the nucleus, which also requires more space. Let's move on. Let's get into, in this one, the importance of DNA. The first thing that I would like to say about DNA is how we use phylogenetics to understand biological aspects in relation to DNA. Phylogenetics, which is the classification of genetic sequences through classification, has a very important significance as it relates to DNA. Nucleotide sequences within DNA, genotypes, which are the biological basis within an organism, help to relate organisms by heredity. Chromosomes can be compared through staining, which highlights significant patterns between organisms. This was the case when investigation of phylogenetics revealed that humans and chimpanzees have similar DNA structures. This meant that these two organisms could share a common ancestor. Not only could this help in establishing ancestral lines, DNA can help reveal the evolutionary differences between organisms, such as the development of gill systems within aquatic organisms. This is very important because DNA is so essential in understanding pedigrees, hereditary information, family trees. Phylogenetics is basically just a categorization of animals or humans, any organism really, but being able to classify it in a organized tree that explains the lineage of how they became the way they are. Right here would be an example of phylogenetics right here, where they have compared skulls of Homo sapiens and Homo sapiens sapiens, and before them, that actually reveal, compared to monkey and chimpanzee organisms, that the structure of the skull was actually similar in history, and the evolution might have something to do with a common ancestor link. Right here, you can see these lines right here resemble common links within each other. So each gap between these organisms represent a commonality between the two, linking them together. Let's continue. HIV. The phylogeny of viruses required the analysis of HIV nucleotide sequences. Although these sequences are similar in shape and structure, the nucleotide sequences are the factor that make them different. Since the 80s, HIV has been one of the leading causes of death among humans and animal species. Finding a cure requires investigation into the origin of HIV and the molecularity behind it. When comparing monkey and human HIV cells in the early stages of research, it was found that the ancestor of the virus did not derive from a pre-existing human virus. This meant that somewhere along the line, a chimp or monkey organism carried a disease that somehow mutated over time and created a deadly virus that was transferable to humans. Right here, I have included a picture of the HIV-1 viron so that you can understand um, how we look at the HIV cell itself and how you can see how it might be different from a human versus a monkey organism or a chimpanzee organism. Crimes are also essential. DNA is also essential in understanding crimes as well. DNA profiles can be very helpful in assessing acts of crime, such as in the cases of serial killers, because things like blood tissue and hair samples can give insight into the victim and the accused. It is essential to understand how to analyze the biological information. Patterns in the victim's death are analyzed, as well as the clues left behind. Police rely on information from scientific databases that can analyze the nucleotides and the sequences of DNA. That is what we're seeing here. This picture right here depicting what nucleotide sequences would look like. This is somebody using the information that they see with the nucleotide sequences to apply to this sample here from a patient, or it could be from anybody really, but this information is what they use to analyze this sample here to see how it lies differently biologically to what they're trying to get, what the point and the basis of this finding is. Police rely on information from scientific databases, which in this case will be DNA profiling. I have a picture down here of DNA profiling, which shows the 3 steps of it and exactly how they use the comparisons to come to a conclusion lawfully. If anyone in the system has been convicted before, they will automatically come up in the system if the DNA sequences match. This is not only true for serial killers and murder suspects, but this is also helpful in finding the person responsible in relation to victims of rape or sexual assault. So, as I said before, you can use hair tissue, um, skin cell samples, anything anything that could be a fluid or anything that um, could be evidence could be used to find and could find the person who did the crime, basically. And lastly, medical significance. DNA also has importance to medicine. DNA can be the answer to treating different medical diagnoses. Understanding the effect of different chemical compositions between organisms and providing answers to open-ended questions such as the significance of the medicine on the body. In the same way the DNA aids in understanding of the AIDS virus, this is the same for infections such as diabetes or heart disease, which can reveal the maternal or paternal link affecting the person's health. If you have a history of heart disease in your family, it will be helpful to analyze the pedigree of your family to determine the likelihood of you developing the same issues. This can be shown through carriers or sex-linked inheritance patterns. Continue. Cell signaling. I feel like this was important to talk about because cell signaling is actually very important to DNA. Without cell signaling within DNA, we would not be able to communicate messages to our bodies to produce reactions, to produce movement, to produce muscle and tissue development. Without cell signaling, I would not even be able to deliver this presentation right now. Let's get into it. Cell signaling is the communication of receptors to cooperate with external stimuli. Most cell signaling relies on pathways of molecules signaling to each other to bring about a response. Some cell signaling pathways include the VEGF signaling pathways, AKT signaling pathway, insulin signaling pathway, apoptosis signaling pathway, to name a few. A signaling cascade describes one molecule signaling to several molecules, which then signal to several more, and so on. This results in an amplified and complex response to one initial signal. Basically, imagine that you have a domino game set up where you have one domino at one end of the room and you have a lot of dominoes all throughout the other room to the other side of the room. Once you press the one domino that starts the ball rolling for the rest of them to tumble down, they will respond to each other and tumble down because the last domino fell, making this one fall. It's all about kinetic energy. As the domino is sitting there, there's potential energy. Cell signaling works the same way. And so the signaling cascade, if you would like a visual representation, you could picture a domino setting just the way I did, where you would knock down one domino and it causes the next to fall, and the next to fall, and the next to fall. That is exactly what happens in the signaling cascade. We are responding with our cells and receptors to get signals across our body to communicate a response. Important factors included in cell signaling are receptors, like I said, ligands, kinases, and transcription factors. The Wnt pathway is the most notable pathway. So the Wnt pathway, signaling pathway, regulates several developmental processes such as cell differentiation, cell migration, and proliferation during embryogenesis and in adult tissues. Dysregulation of this cell signaling pathway may promote tumorigenesis and metastasis. So the Wnt pathway is a very notable pathway whenever you're studying cell signaling, because it has a lot to do with cell structure itself and cell functioning by itself. It actually is one of the pathways that provides the basis before the other pathways can even be established, for your cells to communicate with each other. And without this pathway being established, you would your cells would experience such a great change that it could possibly destroy your cells, which is where tumorigenesis comes into play, where tumors could develop, or metastasis could come into play. So that is the conclusion of my presentation. Thank you so much for your time. Um, I appreciate those who have come in to listen.